right, good afternoon and welcome back. I have to show you this water bottle. This is the coolest. Look at this. It's got a biohazard symbol, so nobody's going to steal it from me, right? And it says "Got Pox" on it. This was from a summer meeting here at Columbia. They gave it to us. So talking about not stealing things, my license plate is viruses on my car. So I applied for a parking permit once, and they say, put your license plate. I put viruses. And weeks went by, and they weren't sending it. So I emailed, and the lady said, oh, I thought it was a virus, so I didn't open it. Gosh. That's why we need to teach the world about virology. Okay, there is water in here, actually. And I hope I don't leave it here, because I really like this orange bottle. Let's talk about viruses making RNA today. And this story begins in the 60s. Let me give you some RNA history. So viruses are discovered, remember, around 1900. By 1935, people started to make crystals of viruses to solve their structures. They were interested in what they were made up of. So this was first done by a guy named Wendell Stanley in 1935. He crystallized tobacco mosaic virus and found that there's only a 5% RNA in the crystals. The rest was protein. So he said the protein is the genetic material. This guy got a Nobel Prize for that. They should take it back, right? 1944, we learned that DNA is genetic material. 1952, the Hershey-Chase experiment, which we talked about, showing DNA is the genetic material of viruses. And 1953, the structure of DNA is solved. By the way, that's the year of my birth. That's why I am a scientist, because big discovery that year. Actually, there are other things that happened that year, too. 1956, RNA in tobacco mosaic virus shown to be genetic material. We talked about these experiments. And then by 1959, many people found RNA in multiple animal viruses. And so they realized this was a thing, that it is genetic material. And so they start to ask, how does this RNA get replicated? Is it the virus? Is it something in the virus, or is it the cell that does it? What we're going to talk about today is shown here in the Baltimore scheme. We're going to remember mRNA is at the middle. That's, of course, RNA. We're going to talk about viruses that have RNA genomes. So we're going to talk about viruses with double-stranded RNA, single-stranded negative sense RNA, and viruses with plus RNA. Now, there's another class of viruses with RNA, but they go through a DNA intermediate, the retroviruses, and they're going to have their own lecture. The first experiments that were done to try and get at this question, what is making the RNA, are like this experiment. In this experiment, you take a cell. This is a cell. It's not a fried egg. It's a cell. And you infect it with poliovirus. And you then make cell extracts at different times after infection. You break the cells open, and that's a cell extract. And then you add triphosphates, the 4 triphosphates. And one of them is usually radioactive. In the old days, we used radioactivity. People don't do that anymore. But we had no choice back then. You add radioactive triphosphate, and then you can measure the incorporation into RNA. So it's a way of measuring RNA synthesis. And the result of this experiment is shown on the right here. So this is a graph. We have hours post-infection, 1 through 6. So very short reproduction cycle. This is poliovirus. And then on the left hand, the y-axis is the RNA polymerase activity. So this is incorporation of the label into RNA. And then on the right y-axis is the titer of poliovirus type 2. So this is PFU per mil, right? So they did a plaque assay here to measure the titer. So those are the square boxes. So you can see between 2 and 3 hours, you have an increase in infectivity. It increases and eventually plateaus. And at the same time, the RNA made increases. Those are the circles. And so you can see there is something is making RNA in these infected cells. And of course, people wanted to know what it was, and it turned out to be viral in origin. There are a lot of experiments that went into doing that, but I'm just going to leave you with the identification of it being viral and not cellular. Many people thought cells could make RNA from RNA, but that turns out not to be the case. All right, so subsequent to that experiment, people found that the enzyme that made that RNA in poliovirus infected cell, we call it an RNA-dependent RNA polymerase, could also be found in the particles of negative-strand RNA viruses. And so that, as we'll see in a moment, the polymerase, the RNA polymerase, has to be in the particle of negative-strand and double-stranded RNAs. For positive-strand RNAs, the polymerase does not have to be in the particle. Nowadays, if you sequence a viral genome, you can identify polymerases by certain amino acid combinations. So here's one, GlyAsp-AsP. GDD is a signature for an RNA polymerase. We'll see why in a bit. And then you can take the gene encoding that protein, you can synthesize it on its own and show that it has polymerase activity. And then you can also solve the structure of the protein. I have actually crystal structures here. That's not necessary anymore. You can do cryo-EM, molecular structure. So that could be crystallography or cryo-EM. Real-time updating of lectures is great. You know, in the old days, we had to make slides, actual physical slides. I don't know if you remember that. And how many people went to a talk and dropped their tray of slides minutes before their talk and had to, oh my gosh. Stories I can tell. All right, so here's a summary of this idea about whether or not you need the polymerase in the particle. So for viruses with negative-strand RNA genomes, the polymerase is in the particle because the negative strand on its own, when it goes into a cell, the cell cannot do anything. It cannot be translated. It's the negative strand. It's the wrong polarity, right? So the polymerase is in the particle. Here's VSV and influenza virus. The polymerases are actually complexed to the RNA. They're part of that nucleocapsid, RNA coated with protein. The positive strand RNA viruses are typically naked. There's no protein coating them. There's no RNA polymerase because the plus strand can go right into the cell and be translated. Now, of course, there are always exceptions in biology, and the two exceptions here are the retroviruses, which are plus strand RNA. They have a reverse transcriptase in the particle because they're going to make DNA from that plus strand. So it does not get translated. And the coronaviruses, which do not have a polymerase in the particle, but they do have a nucleocapsid. So here is the coronavirus, third from the left. The RNA is complex with protein, so it's a nucleocapsid. Turns out the same with retroviruses. They are also complex with proteins. They're also a nucleocapsid as well. So all the other plus strand viruses, the RNA is naked. We're not sure why they're not in these two kinds of viruses. Probably the coronaviruses, the RNA is so big that protecting it with protein is probably needed. And again, the double-stranded RNA genomes, the RNA is naked, there's no protein on it, but there is a polymerase in the particle because if a double-strand RNA gets into a cell, it sounds like a joke, right? If a double-stranded RNA gets into a cell, it's not translatable because the plus RNA in the double strand is blocked by the negative strand. So nucleocapsids we've talked about before as being substructures of a virus particle. Right? They're typically nucleic acid protein complexes that were within a membrane. They can also be within a icosahedral capsid. Here are just two examples of what they look like. On the left is vesicular stomatitis virus, which has a bullet-shaped nucleocapsid, right? The protein, a single protein, nucleocapsid protein, repeated many times. It interacts with the viral RNA and it interacts with itself to form this coil. On the bottom is actually the structure of the single protein and a piece of RNA. You can see there's a groove in the middle of the protein and the RNA is bound in there. And on the left is a structure of a few turns of this nucleocapsid protein. You can barely see this darkly colored area in dark blue. That's equivalent to one of these subunits here. So that's, and of course there would be RNA wrapped up in this as well, but it's not shown there. On the right is the influenza virus nucleocapsid. Here there are 8 of them, 8 RNA protein complexes. And on the right is the structure of one. So we have a nucleoprotein, the orange circles there, bound to each other and to the viral RNA. And then at the one end of this ribonucleoprotein is the RNA polymerase. This is the negative-strand virus, so it has to bring in an RNA polymerase into the cell. And on the bottom is, again, the structure of the nucleoprotein, and you can see an RNA fitting in a groove in the middle of it as well. And on the bottom, a space-filling model of the ribonucleoprotein, and you can see one of the molecules colored dark. So this is a side view, which is kind of analogous to the drawing up at the top there. And then this is we've turned it 90 degrees, yeah, and we're looking down the center, so you can see there's a hole in the center. So those are what nucleocapsids look like. And remember, within these structures, even naked RNA, they're always structured. They have secondary and tertiary structures. These are illustrated here. So here's RNA in green, and even single-stranded RNA can base pair with itself. So you can see we can form stems by having sequences within, not too far away from each other, base pairing. You can have multi-branched stem loops and so forth. So these can get very complicated. You can also have something that's called a pseudoknot, which is shown in panel B. It's called a pseudoknot because it looks like a knot, but it's not. And it's a pseudoknot, you have a stem loop, and then a few base pairs in the loop can base pair with some bases downstream of the loop. That's what the dotted lines mean. And it forms a structure shown in the next panel. So those are the base pairs actually happening. And then it actually twists a little bit. And on the upper right is what the structure actually looks like. So it looks like a knot, but the RNA is not threading through itself and pulling itself as your shoelace would. And so, for example, here's an RNA of HIV here, and it's highly structured. Here's the depiction of this structure. On the top, many stem loops, and that's the actual three-dimensional structure of the RNA. These RNAs are important. These structures are important. They interact with proteins. They have regulatory roles and so forth. They're not just there to look nice. So today we're going to talk about RNA synthesis, and there are rules. So it's RNA-based RNA synthesis. In other words, copying RNA into RNA. And there are rules for that. First of all, that's an RNA molecule. It's in green. So we have color schemes for our molecules in this course. They're taken from the virology text. Green is RNA, and blue is DNA. So that's an RNA molecule. And that's a plus strand RNA molecule because it's a light green. And a dark green is a minus strand. First of all, the first rule is the RNA has to be copied from end to end. You can't lose any sequence whenever you copy it, right? Because then you will eventually not be able to make a virus if you lose the sequence. It's there for a purpose. And secondly, you have to make mRNAs that can be translated by the host cell. And we know this already from the Baltimore scheme, right? That the cellular translation machinery translates these mRNAs, so they have to be compatible with that. And there are a number of features that ensure that. Continuing with the rules for RNA synthesis, typically the synthesis begins and ends at a very specific place on the template. So if you have a template RNA and you're copying it, the initiation is very specific and the termination is. Sometimes it can be the ends, but not always. The synthesis of nucleic acids may or may not need a primer. So on the top, for example, is a piece of nucleic acid called the template, and it's going to be copied by an RNA polymerase, which is that protein in brown there. And some polymerases need a primer, a short oligonucleotide, maybe 10 to 20 bases in length, that tells the polymerase where to begin. So the primer base pairs to the RNA, and then the polymerase sits down and begins to add bases. But not every polymerase needs a primer. And throughout this course, as we talk about different polymerases, I'll tell you when they do or when they don't. Besides the RNA polymerases, you need other viral proteins. You typically need cellular proteins for this whole process. And then the actual polymerization, of course, is based on the template. The polymerase is reading the template. If it sees an A, it will put in a U. right? If it sees a G, it will put in a C and so forth. So that's template-directed incorporation. The product is made in a 5' to 3' direction. So 5' end and the 3' ends of the RNA are shown here. And this primer is shown in the 5' to 3' direction. And the polymerase will add bases to the 3' end only because there's a hydroxyl there that can be used to add more bases. And then it will synthesize in a 5' to 3' direction. But the template is read in a 3' to 5' direction, okay? Here's the template, 3' to 5'. So it's read in a 3' to 5'. Synthesis is always in the opposite direction, 5' to 3'. And so this is called templated RNA synthesis, because obviously there's a template that's directing what the polymerase should make. But sometimes there is non-templated synthesis. For example, we'll see today polymerases can stutter. They can get stuck at a base and just keep churning out bases without a template. And that actually has some role. All right, so let's start with initiation of RNA synthesis. There are two general modes. There's either de novo, which means you don't need a primer. So the word de novo in the context of RNA or DNA synthesis means you don't need a primer. So on the top here, these are the last two bases at the 3' end of the RNA. So the synthesis will begin at the 3' end, and the polymerase can just start adding NTPs that are complementary to those. And then we have cases of polymerases that need primers. They will not copy the template without a primer. Some polymerases utilize primers that are linked to a protein. We'll see an example of that today. And some polymerases utilize primers that are capped. So cellular mRNAs have a cap at their 5' end. It's a chemically distinct group, which we'll talk about later. And some polymerases require a capped short oligonucleotide to use as a primer. We'll see examples of these today. There are universal rules actually for both DNA and RNA synthesis that are based on this mechanism discovered many years ago. It's called the two-metal mechanism of catalysis. You may have encountered this in biology courses, but here it's shown how it works. Here's a template strand on the right showing all the detailed atoms. So here's the 5' end, and then we have a series of bases and the 3' end. And of course, when I say bases, it's not entirely correct. The base is just the T or the A or the G or the C. The base is linked to a ribose, and then the ribose is connected to the next base by a single phosphate linkage. And these bases, of course, come as triphosphates. Those are the precursors for polymerization. And here, for example, so here is a T being added to this A by the polymerase. And you can see it's got 3 phosphates, 1, 2, 3. So it's a TTP, thymidine triphosphate. But during the polymerization, the last 2 phosphates are removed, and you end up with just 1 phosphate in between the bases. So here's where the two metals come into play. In the polymerase, which we'll see in a moment, there are amino acids that hold in place 2 magnesium ions. And they're typically aspartates for RNA polymerases. So you can see aspartate A and C here. They are holding those magnesiums in place. And the magnesiums are needed to help catalyze the reaction of adding a new base. So in this particular synthesis, we're synthesizing 5' to 3'. So the T is the next base to be added. We have a hydroxyl typically at the end of the last base there. And that oxygen will attack this particular phosphodiester bond and cause that phosphate to attack the oxygen, and then the 2 phosphates are released. And all of that is coordinated by these 2 magnesiums, which you can see are forming non-covalent interactions with the oxygens here, 1, 2, 3, 4. So the magnesiums hold these bases into place so that this catalysis can happen. And of course, the polymerase helps this catalysis proceed by lowering the energy requirement. So that's the two-metal mechanism, and we'll see how this actually works physically today when we look at the structures. So our first question today is, which is a universal rule about RNA-directed RNA synthesis? The RDRP, RNA-dependent RNA polymerase, may initiate de novo or require a primer. RNA synthesis initiates randomly on the template. It's synthesized in a 3 to 5' direction. RNA synthesis is always template directed. So which is a universal rule? All right, let's see how we did. So most of you got the right answer. The correct answer is RDRPs may initiate de novo or require a primer. That's correct. Initiates randomly, no, always initiates specifically on the template. Nobody got that, that's good. It's synthesized in the 3 to 5' direction. No, it's synthesized in a 5 to 3'. The template is copied in a 3 to 5' direction. It's always template directed. No, it's not, and I'll show you the slide where I said that. There is some non-templated synthesis, which we'll actually see today. Now, all the structures of the 4 different kinds of nucleic acid polymerase have been discovered. So what are they? So today we're talking about RNA-dependent RNA polymerases. And so here's a structure of the poliovirus polymerase on the right. And then the other 3 kinds of polymerase, there's reverse transcriptase, which is RNA-dependent DNA polymerase. There's DNA polymerase, which is DNA-dependent DNA polymerase. And then, sorry, this T7 RNA polymerase is actually DNA-dependent RNA polymerase, the one that makes messenger RNAs. And then finally is DNA-dependent DNA polymerase. So these are the 4 classes of nucleic acid polymerase. RNA-RNA, RNA-DNA, DNA-RNA, DNA-DNA. As far as we know, there aren't any others than that. And the structures of many of these have been solved, and they all look very similar. They look like a right hand with fingers, thumb, and palm domains. The fingers and the thumb kind of encircle the active site, which is the palm domain shown in that picture as a little red dot. And we'll explore that in a moment. On the top are the amino acid alignments of these 4 classes of polymerase. And the colored areas are similar sequences between the different polymerases, the most similar sequences. As you can see, this motif A is in red. It's conserved in all of the polymerases that you can see. And so the green and the yellow and the purple and so forth. And so we think these probably originated from a common ancestor, maybe most likely an RNA-dependent RNA polymerase. But of course, that's a long time ago, pre-cellular, so we don't really know. The yellow domain is where the catalysis occurs. So the yellow domain is in the palm. And here you can see in the polio polymerase 2 beta strands that are making the yellow domain. That's where catalysis happens, the same for all the other polymerases. And in that yellow domain, the active site, that's where there are amino acids that are crucial, that hold those magnesiums into place, right? I told you about these before, like gly, asp, asp. The 2 aspartates coordinate those 2 magnesiums to allow the catalysis to happen. And you can see in other viruses, you have gly, asp, asp. Sometimes you have ala, asp, asin, little variations on each one, but that's the function of this triplet is to coordinate those magnesiums. So there's some conservation, obviously, among these. So here is a closer look at the poliovirus RNA-dependent RNA polymerase. Again, it's a right hand. This is the fingers domain, and the thumb on the right. They're touching. For polio polymerase, they touch. They don't touch for all the polymerases. Sometimes there's an open hand configuration. And again, you can see the yellow, the 2 beta strands that constitute the active site. And these 2 amino acids where the side chains are shown as balls and sticks, those are the 2 aspartates that are coordinating those magnesiums, and they're completely required for catalysis. If you change one of those to something else, the polymerase is inactive, just 1 amino acid change. So that's where they're located, and that's where the catalysis happens. So let me show you a different view so you get an idea of how this works. So these are now space-filling views where we have all the molecules as spheres against the polio RDRP. And the yellow, again, is the active site where we have those 2 aspartates. And in fact, the 2 aspartates are shown here in purple. And the template enters on one side of the molecule. So here's a top view of the polymerase. We're looking down. We've removed some amino acids here in the front so you can see inside. The RNA comes down the top. It passes over the active site. It comes in single-stranded, and then it's made double-stranded. And then the double-stranded RNA exits. So you can see here the green initially came in. Well, actually, on this picture, they're putting blue in. It should have been green, but it's single-stranded blue. And then the second strand that's made is green, and the double-stranded RNA exits. Here's the front view. So you can see the single-stranded template going in the top. It turns to a 90-degree angle and comes out double-stranded on the front. So the top and the front are where the nucleic acids enter and exit. And of course, as they're entering, they're passing over the catalytic site and they're made double-stranded. There's one other important part of this polymerase, and that's the back. And the NTPs actually enter the back. So if you go in the back, you have access to the active site, which it's yellow here. So the NTPs come in the back. It's a big enough channel for that, and then they're put in at the active site. The way this works actually is, let's say there's an A at the active site that needs to be polymerized next, and you need its complement, which would be a U. There's no way for the enzyme to know. How does it know to get the right triphosphate? It doesn't. It tries all four. So there's some triphosphates floating around, and they randomly come in, and if it's the wrong one, it leaves. It doesn't have a high enough affinity. And the right one finally comes in, it base pairs, and it's polymerized. So that's happening really, really fast, but it's a random thing. It's not like the polymerase says, it's an A, give me a U. No, it doesn't happen that way. They're all four in there, and it's trial by error, trial and error. Now, I want to go through this process of RNA synthesis for plus, minus, and double-stranded RNA viruses. So for plus, we're going to look at polioviruses and also alphaviruses because they're slightly different. So poliovirus is the genome is a plus-stranded RNA, and it is copied into a negative strand, which simply serves as a template to make more plus strands. This negative strand does nothing else except to serve as a template. Seems like a waste, but that's what you have to do. These are both genomes and messenger RNAs. So for flaviviruses, West Nile, dengue, the RNA is actually capped at the 5' end, but for poliovirus, it's not capped. For the alphaviruses, chikungunya virus, Sinbis, they're also plus-stranded RNA viruses, they're capped. And by the way, you see they're also polyadenylated at the 3' end. They're bona fide messenger RNAs. These viruses also synthesize a subgenomic RNA. In other words, an RNA that's not the complete plus strand, and uses that for making proteins as well. All right, so let's take a look at this in some detail. So poliovirus binds its receptor, which we talked about last time. It's taken in to the cell. The receptor catalyzes the formation of a channel in the particle and the membrane, so now the RNA is in the cytoplasm. Since it's plus-stranded RNA, it can immediately be engaged by ribosomes. The ribosomes make proteins. These are actually made as one long protein for this virus, which is chopped up by proteases encoded in the genome. In fact, that's how the coronaviruses work also. And Paxlovid, the antiviral, inhibits one of those proteases. Among the proteins that are made include the polymerase, which copies the plus RNA into double-stranded RNAs, a plus and a minus strand, and then the plus strands come off and are packaged into new virus particles. So very simple scheme, plus to minus, back to plus. And this replication happens on vesicles, membrane vesicles that are induced by virus infection. We'll talk a little bit more about these later on in the course. This is a schematic of the viral genome. So you can see the RNA at the top. It's about 7,400 bases long. There is a protein linked to the 5' end, and not a cap, but a protein called VPG. There's a poly A at the 3' end. And when ribosomes engage this RNA, they make a very long protein, which is called a polyprotein. And then it's processed to the individual proteins by two viral proteases. And you get capsid proteins and the rest of the proteins involved in RNA synthesis, including the polymerase, which is here 3D Pol. At the very 5' end, the protein is linked to the RNA by a phosphodiester linkage to a tyrosine. So tyrosine is one of the amino acids that has a hydroxyl, right? So that hydroxyl can be linked to the phosphate of the first base. And you can see that here. This is a uridine. So phosphodiester bond. And then the rest of the genome is shown here. There's a cloverleaf structure here, very at the 5' end, which is involved in RNA synthesis, as we'll see, and then the rest of the genome follows. So that's a covalent linkage that occurs between the viral protein that is linked to the RNA. So this is another kind of view of the viral RNA, which shows some important secondary structures. Remember, I said secondary structures happen in RNA, and they are functional. So for polio RNA, we have a clover leaf at the 5' end. We have a stem loop somewhere in the middle. It doesn't actually matter. It varies where it is. And this is called a Cre element. And then at the 3' end, we have a pseudoknot. These are all signals for RNA replication that distinguish the viral RNA from cellular RNAs. When polio infects a cell, the polymerase does not copy any cellular RNA whatsoever. It only copies viral RNA. And it's because the viral RNAs only have these signals in them. So to start RNA synthesis, that Cre element that I just showed you, it actually serves as a template for the polymerase, which is shown here as 3CD. Polymerase binds to this Cre element, and then it takes a free molecule of VPG, which is the protein that's going to end up at the 5' end of the genome, and it adds 2 U residues to it. UU, which is going to be the primer for RNA synthesis. So you could guess the first 2 bases of the RNA are AA. They're going to base pair with that UU. Those U's are added because this loop in the Cre element has a series of A residues, so it's a templated reaction. Okay, now we have a primer for RNA synthesis, and that primer is going to sit down on the RNA. So here's a viral plus strand RNA that's about to be copied by the polymerase. The polymerase is here's the Cre element in the middle. So the polymerase is uridylated VPG, adding 2 U's. And somehow it gets from the Cre element to the 3' end of the genome, which is shown there, the poly-A sequence. And then the polymerase will start to elongate from that primer and eventually make a full-length minus strand of the whole genome. So that's the primer step. Now, this whole affair occurs on membranes. This is a cellular membrane at the top left. And in addition, the RNA is circularized. So the 5' and 3' ends are brought together by protein-protein interaction. So there is a protein in the cell called poly-A binding protein, PABP. It binds poly-A. It also binds a molecule of the polymerase that is bound to the clover leaf. And that interaction requires the clover leaf to be bound to a cellular protein called PCBP. So all of that does is to circularize the RNA, brings the 5' and 3' ends together. So this polymerase that's bound to the clover leaf is the one that's going to start copying the viral RNA. And it does so and makes a double-stranded copy. This process is carried out, as I said, on membranes, those membranes that are induced by virus infection. We'll talk more about those, I think, in lecture 11. Our next question is, which is a part of the poliovirus replication strategy? The production of subgenomic mRNAs, de novo without primer initiation of RNA synthesis, circularization of template for initiation of RNA synthesis, all of the above, which is a part of the poliovirus replication strategy. Now you may be thinking, why does anyone need to know any of this stuff, right? Paxlovid is a protease inhibitor. We wouldn't know about the ability to inhibit a protease if we didn't know this kind of thing. And there are other antivirals that inhibit polymerases, which you need to know. There's an antiviral that inhibits an endonuclease that's involved in influenza RNA synthesis. So it's only when you know the basics that you can really devise antivirals and even vaccines that work. So if you're a doctor one day and you give your patient a prescription for Paxlovid, and they say, what is an M protease, which is the target? You're going to know because of this course. they'll think you're really smart and they'll keep coming back to you, which is what you want. Okay, let's find out how we did. So the right answer is 52% of you. Circularization of the template. You don't make subgenomics. It's not without a primer. Nobody picked that. It's not all of the above. It's just circularization. So let's have a look. Since not everybody got that, let's take a look at what's going on. So there's circularization of the template, right? There's interaction between poly binding protein and the polymerase, brings the 5' and 3' ends together. That's what that's all about. And so it is a primer-dependent reaction, and then you make full-length copies of the plus strand, which are the minus strands intermediates. The other plus strand virus we'll mention briefly are the Alphaviruses. These include chikungunya virus, which you may have heard of because the name is so cool. But it actually causes an arthritis-like disease, and it's mosquito-borne. And these viruses make a subgenomic RNA. That's why I want to tell you a little bit about them. So these viruses bind receptors. They're envelope viruses. They are taken into endosomes. pH drops. You have fusion catalyzed conformational changes, and the RNA ends up in the cytoplasm. So now it's a plus-stranded RNA. There's no polymerase in the particle. So that's translated directly to make a single protein, which is the RNA polymerase. Because you got 4 subunits to it, but that's the RNA polymerase, this NSP 1, 2, 3, 4. So that RNA polymerase will then copy the incoming plus RNA to make negative-strand RNAs. And then some of those negative strands are going to be copied to make a subgenomic RNA. So the input RNA is not fully translated. To get to the rest of it, you need to make a subgenomic RNA, and that subgenomic encodes the structural proteins, the capsid protein that makes up the capsid shell, and then glycoproteins that end up in the membrane of the virus. So here's a schematic of what's going on here. Here's the plus-stranded RNA. When it gets in the cell, the first half or so is translated to form the polymerase protein. And there's a termination codon here in the genome that stops translation. And then the polymerase makes the negative strand, and then the polymerase makes from the negative strand a subgenomic mRNA. So subgenomic simply means it's smaller than the genome, right? So here it is. It's the right-hand half of the genome. The red arrow is the initiation site for synthesis of that mRNA, and that is then translated into proteins. Now you may say, what's the function of this? Poliovirus basically makes one long polyprotein. Why isn't it the same here? It works, right? Evolutionarily, it's competitive. And this is just another bifurcation on the evolutionary pathway. You can have a genome be translated completely, or you can make a subgenomic RNA. That's the only thing I can tell you about why that happens. Now, coronaviruses are a twist on this strategy. They are plus-stranded RNA viruses. They do not have a polymerase in the particle because the RNA is plus-stranded, but they make lots of subgenomic RNAs. So in a coronavirus-infected cell, so there's a coronavirus binding to its receptor. Some of them fuse at the surface, remember, after the spike protein is cleaved. Others can fuse within endosomes. Either way, the RNA is in the cytosol. The first third of it or so is translated to make the polymerase enzyme. And that polymerase can either reproduce the genome to make full-length genomes that you want to make new particles with, and we'll talk about assembly of particles separately later, or it can make mRNAs. And the polymerase will take the plus-stranded genome and make negative-stranded mRNAs, and then from those it will make plus-stranded mRNAs, which are then translated to make a variety of structural proteins. And these mRNAs are unusual because they're nested. In other words, you have a big one and then a smaller one within it and a smaller one. Nest in Latin is nido. It's the name for the order of viruses that includes the coronaviruses, Nidovirales. That's why. And the nested nature is shown on the right. Here on the left is the genomic RNA. And these boxes are all the proteins that are encoded. Probably you recognize S, the spike protein, right? Because it's the basis of all the vaccines against SARS-CoV-2. But there are other proteins as well. When this RNA comes in the cell, The left half is translated to make the polymerase, which then goes on to copy the genome, as I've said, and you end up with nested subgenomic RNAs. And this is very interesting because the biggest one encodes all of these proteins to the right of the polymerase, but only the first one is translated. The ribosomes bind to that RNA, the first protein. So here 2A would be translated, here HE. This is the spike mRNA, and all the way down to N, the last protein, the nucleocapsid protein, is made from a tiny mRNA that only encodes that protein. And if you ask what's the function of this strategy, can't give you a good answer. It just exists and it works. How it works is very interesting. Here's the genome at the top here, and it contains a number of intragenic sequences. The polymerase will recognize the 3' end of the plus strand, and remember, it can make long minus strands, or it can make subgenomic RNAs. And the polymerase begins to initiate synthesis here at the 3' end. And then at this first region, it will stop and make one particular subgenomic mRNA. And it does so by copying and stopping, going a little further and a little further. And that's how you get that nested set of mRNAs. And these are all negative strands initially, and then they're copied to make the plus-stranded RNAs. Now, what's unusual here is that there is a leader sequence, which is shown here in gray and blue, which is added to each one of these mRNAs. And the mechanism is that the leader sequence is encoded at the very 5' end of the genome. And so for each mRNA to have that leader sequence requires the genome wrap around and be copied by the polymerase in this kind of configuration. So here's the linear version. Polymerase is copying. When it gets to this intergenic sequence, TRSB, apparently the RNA is folded around, and then the polymerase copies from the 5' end of the same molecule. It does that for each of the mRNAs that are made. And the reason I'm telling you this is because this is a kind of recombination. When the polymerase changes template, actually not template, but it's going from the 3' end to the 5' end. And this ability is why coronaviruses have high recombination rates. If you look at the SARS-CoV-2 genome, it is a mosaic of many different SARS-CoV-2-like viruses because recombination occurs at high rates, and we'll probably talk about that later. Minus strand RNA viruses, here the problem is you cannot be translated, so you have to have the polymerase in the particle. So here the negative strand RNA is shown in the darker green. We're going to talk about viruses with one RNA, unimolecular like VSV, and we're going to talk about viruses with segmented genomes like influenza virus on the right. In both cases, the strategy is going to be the negative strand RNA comes in with the particle with a polymerase. And then it is either copied to form mRNAs or it is copied to form full-length minus strand genomes to make new particles with. So let's go through that for VSV. VSV is one strand of negative stranded RNA. It's not messenger RNA, right? It's negative strand. So it has to come in with a polymerase. The virus enters by endocytosis. The RNA protein complex is released into the cell. The polymerase on the genome copies it to make subgenomic mRNAs, as shown here. Those are engaged by ribosomes, and they encode for a variety of proteins. And among those proteins, structural proteins to build new virus particles, and of course, the RNA polymerase that is going to take that negative strand, it's going to make a plus-stranded copy of it, and then it's going to copy that back to new negative strands, which could either go into new particles or be recycled into making new proteins. Now, there's a special situation here, and that is when the viral genome is not mRNA, there has to be a switch from making mRNA, which happens early in infection. When you need a lot of proteins to be made, you need to make mRNA. You have to switch from that to later in infection, making a lot of genomes. And we're going to look in these cases that we go over where that switch is occurring. And for a virus like poliovirus, where the genome is mRNA, there's no switch. It doesn't have to be because it's the same molecule. But here, there's a switch because the mRNAs are different from the genome. So here is the viral genome for VSV. Remember, bullet-shaped particle related to rabies virus. Negative-stranded RNA, so no cap, comes in it's in the virus complexed with the nucleocapsid protein and a molecule of RNA polymerase, which consists of two proteins, L and P. And as soon as that RNA is released into the cytosol, the polymerase makes subgenomic mRNAs from it, 1, 2, 3, 4, 5, that encode all of the viral proteins. So the mRNAs are made, they're translated in the cytosol, and here L is the polymerase protein, G is the glycoprotein spike, and so forth. So that's the overall strategy. How does it happen? So here again is a look at the negative strand here in the middle, minus strand genome RNA. It's coated with nucleocapsid protein, as it comes out of the particle. The polymerase is attached to the 3' end. It will copy this negative strand, and it makes 1, 2, 3, 4, 5 subgenomic mRNAs. What it does is the polymerase starts at the 3' end, it makes the N mRNA, it stops, it then starts again, it makes the P, stops and starts again, and so forth, all the way down to the very end. At some point in replication, you have to have a switch from mRNA synthesis to making genomes, because towards the end of a cycle, you need to make viruses, you need genomes for that. How do you do that? So you have to have some way of making full-length plus strands as the intermediate. These mRNAs cannot serve as an intermediate. They're pieces and they're not full-length copies. So the polymerase switches modes. It begins to make full-length plus strand. And the switch is controlled by the nucleocapsid protein, the N protein. As you make more and more N protein in an infection, the nucleocapsid protein coats the plus strand RNA that is being made, and it forces the polymerase to keep copying without terminating and making individual mRNAs. So the N protein acts sort of like an anti-terminator. The mRNAs are made by terminations, start, stop. Those are eliminated by the nucleocapsid protein. Now you get a full-length plus strand, and the polymerase copies it to make a full-length minus strand, which is also coated with nucleocapsid, of course, because it's going to go into the particle. And here's a look at how this is happening. Here's our negative strand at the top. There's the N gene, the P gene, the M gene, and it goes on. There are intergenic regions that have the start-stop signals for the polymerase. The polymerase is here at the left. It initiates, it makes the N mRNA. Then the polymerase stops at the intergenic region and starts again and makes the P mRNA. So there are these stop-start sequences of events. And again, to make a full-length plus, you just need enough N protein to antagonize those starts and stops. The other thing that happens at this intergenic region is some non-templated RNA synthesis to make the poly-A tail. So here is the end of an mRNA being made. This is an intergenic region where we have all of these bases shown. So there's the RNA polymerase. It hits this U stretch. There's a stretch of 1, 2, 3, 4, 5, 6, 7 U's here. And the polymerase stalls and begins to stutter. It just keeps cranking out A's without moving until it's added about 200 A's, and it stops doing that and it moves on to the next gene. And that then releases an mRNA with the poly-A tail. So the poly-A tails of these viruses are not templated. They're added by non-templated RNA synthesis, where the polymerase encounters this stretch of U's, begins to stutter. We call it stuttering. We add lots of A residues and then stop at about 200. And then the polymerase will reinitiate at the next mRNA and do the same thing over and over. And so all right, so that's VSV. A similar event happens in influenza virus infected cells. You have those events, but on a scale of 8 different mRNAs, 8 different RNA segments. So the genome is negative stranded, but it's in 8 pieces. And again, when the virus infects the cell, we saw the other day how the RNAs get out. They actually go in the nucleus. where they are copied to negative strands, and then the negative strands are copied into mRNAs. The mRNAs are exported to the cytoplasm to form virus particles. And we'll talk about how that works later, but today I just want to focus on the RNA. And again, there's a switch from mRNA to genome because the genome is not mRNA, it's a negative-stranded RNA. So this is what the genome looks like. 8 segments of negative-stranded RNA. From them, 1 or 2 messages are made. For most of them, 1 message is made. From 2 of the segments, the primary mRNA is actually spliced to give a different RNA that encodes a different protein. But essentially, the same series of events happens on each segment that we're going to talk about now. And this should look just like VSV with a few twists. Here's the negative strand RNA that's coming into the cell. The polymerase copies it to make an mRNA shown at the top there. And the mRNA, of course, is used to make protein. Now, this synthesis is very unusual. First of all, it is primer dependent. And the primer is a capped fragment of host mRNA. Our mRNAs are all capped, and there's an enzyme in the influenza virus polymerase that cleaves our mRNAs to yield a cap in about 12 or 13 bases, and that's the primer for influenza virus RNA synthesis. So every influenza virus RNA has some sequence at the 5' end which is non-viral. It's not coding, but it's non-viral. And the enzyme that does that, the endonuclease, is the target of a brand new antiviral, baloxavir, for influenza treatment, which is actually better than the neuraminidase inhibitors, but we'll talk about that later. And at the end, the RNA is polyadenylated, but it falls short of the end of the RNA, so it's not a complete copy. And so at some point, you have to switch from mRNA synthesis to full-length plus strands because the mRNAs are not suitable for copying to make genomes. And again, the nucleocapsid protein is the key for that. When enough nucleocapsid proteins are present in the cell, the polymerase will no longer make mRNAs. It switches to making full-length plus copies of the negative strand, and those can be copied into negative strands to make more virus particles. Okay, so the mRNA is not the same as the genome, so we have a switch. The switch, again, is controlled by nucleocapsid protein. The priming event that I talked to you about is initiated by this endonuclease. So here's the negative strand RNA of the virus. Those are the last bases. And the endonuclease of the virus takes a cellular message, which has a cap on the 5' end, and then a number of bases, and cleaves it about 13 bases from the end. And then that's used as a primer for RNA synthesis. So you can see the mRNA that's made by the polymerase has about 13 bases plus a cap from host cell mRNAs. And there are a couple of other viral RNA polymerases that do this as well. And again, the enzyme that does this cleavage is inhibited by an antiviral baloxavir. Polyadenylation of these mRNAs is also similar to that of VSV with a twist. So on the top is the polymerase complex that is actually copying these mRNAs. It's got 3 subunits, PB1, PB2, and PA. The 5' end of the RNA, the viral RNA, binds to a specific site on the polymerase there, shown with red. And then the polymerase, of course, starts copying at the 3' end, and it pulls the RNA through the protein. So the protein doesn't move down the RNA as you would think it might. But the RNA is attached to the polymerase, and then the 3' end is drawn through the active site, and then the mRNA is made. And that's shown here, capped and coming off. There's a stretch of U near the 5' end of the viral RNA, as you can see there. And when the polymerase gets to that, it can't pull through it, right? Because the 5' end is attached. So it's like a string tied down. So the active site has a stretch of U's in it. And of course, the polymerase begins to synthesize a bunch of A's until that mRNA is polyadenylated. So it's a sort of similar mechanism for polyadenylation as VSV, although the details are different. But again, an example of non-templated RNA synthesis. There's just 7 U's, and you end up with 200 or so on the mRNA. So that leads to the question, how are influenza virus and VSV RNA synthesis similar? So A is the switch from mRNA to genome synthesis is controlled by an RNA binding protein. Polyadenylation occurs at a short stretch of U. Viral mRNAs are shorter than minus genome RNA, or all of the above. Which are similar? Let's see what we did. So most of you got all of the above, which is right. The switch is controlled by an RNA binding protein, which is NP. Polyadenylation occurs at a short stretch of U, and the mRNAs are shorter than the genome, right? For VSV, they're all those tiny subgenomic mRNAs. They're clearly shorter for flu. It is short of the end of the genome. Let me show you that. Here, the mRNA is about 20 bases short of the 5' end of the genome. So that can't be a template for viral RNA synthesis. So in both cases, the mRNAs are shorter. So for VSV, they're clearly shorter, right? Influenza more subtle. And finally, let's talk about double-stranded RNA viruses. Reoviruses, you have a double-stranded RNA in the particle, no polymerase. I'm sorry, there is a polymerase in the particle, but not complex to the RNA. The polymerase makes a plus strand that is translated into protein, and it makes plus strands that are eventually copied and used for new genomes. So let's take a look at how that happens. So there are 3, 6, 7, 8, 9, 10 double-stranded RNAs for reovirus. These viruses can have different numbers of double-stranded RNA. And the particle comes in, it enters the endocytic pathway, the pH drops, and there are also proteases in the endosome. And those proteases strip off the outer shell of the virus. Remember, there are 2 icosahedral shells. And what ends up in the cytoplasm is a core. So the core is hydrophobic and can push its way through the endosome membrane into the cytosol. So the core has the double-stranded RNAs in it. And the core also has the RNA polymerase. The RNA polymerase in the core begins to synthesize mRNAs. The RNAs never leave the core. They remain in there. It's one example where the RNA is not in the cytosol. And those RNAs come out from each 5-fold axis. There is a kind of turret structure at each 5-fold axis, which is open to the interior. And that's plugged by the outer shell in the intact virion, so it's not accessible. But in the cell where you have this core, the mRNAs can come out, and those go into the cytosol. They're, of course, translated into protein. They can be used to assemble new virus particles. So here in step 8, we have a viral particle being assembled, which has mRNAs in it. That particle also has polymerase, so that polymerase can make those double-stranded, and eventually the other proteins can be added, and you have a mature virus particle. So the function of the outer shell is to keep those turret-like structures plugged and to cover the hydrophobic parts of the core. And then you strip off that outer shell. The core is now hydrophobic and leave the endosome. And then the mRNAs made in the core can go out through the openings at the 5-fold axis. So where's the switch to genome synthesis? It's here in the newly assembled core, where these mRNAs no longer can be translated when they're in the core, right? So as soon as you put them in the core, you're now switching to genome synthesis, because in that core, the polymerase will copy them and make them double-stranded. So that's where the switch occurs. This is a schematic of the genome. Should have showed you this earlier. The double-stranded RNAs each encodes an mRNA that encodes one or a few proteins. And here's the virion, and then an intermediate where the outer proteins are removed, and then all of the outer proteins are removed to give you the core particle. And that's the one where the mRNAs can be made and they can come out from these turrets. Now, here's a cryo-EM structure of a core, of a reovirus core, which is assembled in the cell in the process of making RNA. And so you see RNA molecules exiting at each 5-fold axis. That's where the turret would be. You don't see it on this reconstruction. And the idea is that there's a polymerase underneath each 5-fold axis that is synthesizing the mRNA from the double strand, and then the mRNAs go out, and those mRNAs are shown here with caps, and they're in gray. All right, finally, last topic for today. RNA viruses are unusual. They undergo a lot of mutation and recombination. Where does that come from? First of all, mutation comes from misincorporation of nucleotides by the polymerase. So all polymerases make errors, but RNA polymerases don't have error correction mechanisms to remove the error. And so the error frequency of a polymerase in an RNA virus is one mistake in 10,000 or 100,000 bases. So in a 10,000 base genome, a mutation frequency of 1 in 10,000 means one mutation per genome. So in any cell where you have tens of thousands of viruses made, every position can be mutated. The DNA viruses are different, as we will see later. They have error correction to get around this, but RNA viruses do not. There is an exception, though, that Virales, which includes the coronaviruses, have a protein that is error correcting. It's called XON. It's an exonuclease, which basically looks for mismatches, the wrong base, and it cuts it out. So this it gives you about a 15 or 20-fold decrease in mutation rate, because if you remove this protein, you get a virus with a 15 to 24-fold increase in mutation rate. And we think that these viruses have an error correction protein because the genome is big. It's not the right order. They're big because they have error correcting protein in them. Genomes can be up to 40 kb in length, and no other viral genome is that big because they don't have error correction mechanisms. If you don't correct errors, a genome of that size will simply sustain too many errors on each replication, and it won't be viable. Now, for poliovirus polymerase, it's been possible to identify amino acids that control fidelity of polymerization. In other words, the incorporation of the wrong base. That is completely controlled by how the template, the primer, and the NTP interact at the active site. So here's a structure of a base interacting at the active site. Here are two aspartates in yellow, 328 and 329. Remember, they're coordinating the magnesiums. And this is a base that's just come in. It's hydrogen bonded with surrounding amino acids, and it will be added by the polymerase. And remember, I said that all 4 NTPs come in and out of the active site very quickly. So what makes the one win? Well, only if it's correctly base paired does it transmit a conformational change to the rest of the protein. It reorients the triphosphate and allows phosphoryl transfer to occur. Here's the triphosphate here. So there's the ribose ring, and the rest coming off is the triphosphate. If it's the right base, that triphosphate will be reoriented by the polymerase, and it will be incorporated into the chain. If it's the wrong base, it will not reorient, and it will fall out, and the next one will come in. So that's how it samples the bases continuously. The right one induces the right conformational change for polymerization to occur. Pretty cool. Now, a single amino acid in the poliovirus polymerase has been found that regulates the fidelity of copying. And this is amino acid 64. It's a change from G to S, glycine to serine. If you make that one amino acid change in the polymerase, it makes fewer errors. It makes fewer errors. And we think this is a remote from the active site, if you can see here. There's the active site in yellow, and this is on the back of the molecule. And we think this change slows conformational changes, that the ones that occur on NTP base pairing. So if that happens very quickly, you may make a mistake, and this position somehow regulates that. So it makes the enzyme more dependent on correct NTP base pairing in the active site. And so basically, if you change G64 to S, it becomes less good at finding the right triphosphate in the active site, and it makes it better at finding the right conformation, and you make fewer errors. But as we'll see when we talk about evolution, these don't occur in nature. Viruses don't have better polymerases. They all have polymerases that make mistakes. It's evolutionarily beneficial to make errors. That's how you evolve. And we make errors too. Our DNA polymerases, they make 10-fold less error than RNA polymerases, but they still make errors. And that's why we get diseases, but it's also why we can evolve as a species and why all living things can evolve, because their polymerases make errors. Now, another contribution to diversity of RNA virus is recombination. RNA can recombine, and what happens is polymerase copying a particular RNA, let's say it's copying the RNA here called the donor, it can get to a certain point and then switch copying to another RNA. So then in the end, you have a recombinant RNA that consists of sequences from both genomes. So this is exchange of nucleotide sequences among different RNAs. It's different from reassortment, where segmented genomes can mix in an infection. And this is always creating new genomes in the RNA world, and it can be very high frequency. For poliovirus, it can be up to 20% in a single replication cycle. And again, we've identified sequences in the polymerase that regulate RNA recombination. And there are 4 amino acids, not 3. You can see they're listed here, but their names are not so important for you to know. 4 amino acids regulate recombination frequency. So here's G64, right, which regulates fidelity, the incorporation by the polymerase. And here's the active site. And here are 4 amino acids all in a row here in cyan, green, red, and brown. They all control recombination frequency. So that's located in the thumb domain, and it's the RNA exit channel. And what we think is happening is that if I showed you where the primer is in this structure, the primer would be aligned along these 4 amino acids. So we think that these amino acids in their wild type state allow the virus polymerase to distinguish between whether the right RNA is there or not. And so you end up with a base level of recombination frequency. And if you change those amino acids, you can decrease the recombination frequency. So we'll see some implications of this later on. But those are 2 ways that the RNA virus world undergoes change: by mutation and by recombination. And they're both regulated by the RNA polymerase. Okay, so that is RNA-dependent RNA synthesis. Next time, we're going to talk about another kind of RNA synthesis where the template is DNA.